Autophagy is a process by which cellular components are directly uptaken by lysosomes and degraded in the lysosomes, and the reuse of the components are occurred in the cell. Hello friends, welcome to Biology Concern. I am Animesh, and in this video I am going to discuss about the autophagy, what is the autophagy and the types of autophagy. So let's jump into the topic. First of all, the autophagy is a highly conserved eukaryotic cellular process. So basically it is a eukaryotic cellular process which is particularly a recycling process through the degradation of cytoplasmic organelles, proteins and macromolecules, and the recycling of and the breakdown of the products. So basically when a protein or any macromolecule in the cell are formed differently, that means it is defected in nature, then this term autophagy or this process autophagy initiated within the cell. So basically by the autophagy, cell basically reuse of its components, that is let's take an example for proteins. So proteins are basically made up of amino acids. Okay, so whenever a defaulty proteins is produced, then autophagy is initiated, and by the process of autophagy, lysosome breaks down those polypeptide chains and form a unit, that is the amino acids. So each and every amino acids are separated from the polypeptide chain, and thus the cell reuses those amino acids and produces to make another protein. That is the usual action of the autophagy, that is the reusing of its components. So autophagy plays an important role in cell survival and maintenance. Also autophagy lead to the degradation of cargo, whatever it is, that is the macromolecules. It should be a macromolecule, proteins, etc. So and the breakdown of these products back into the cell for the reuse by the cell. Okay, so autophagy is divided into three major categories or three different types. First one is the macro-autophagy, second one is the chaperon mediated autophagy, and third one is the micro-autophagy. So what about the macro-autophagy? So in the case of macro-autophagy, cell produces a double membrane structure, but this double membrane structure formed within the cell, not from the outside of the cell, and those components are fused with the lysosome directly and produces autophagosome, and those autophagosomes helps to digest the product present within this double membrane component, or critically we also called as autophagosomes or endosomes. So endosomes are formed within the cell, and the components that is realized within these endosomes are getting digested by these lysosomal hydrolases. So the lysosomal hydrolases which is present inside the lysosome. So basically whenever an endosome is produced inside of a cell, so basically these endosomes are double membrane compartments, and those double membrane compartments of endosomes are fuses with this particular lysosome, and then these lysosomal hydrolases released from the lysosome, and it basically engulfs the particular components present within these endosomes or autophagosomes, and it specifically digested those products, and the permeases that is present in the lysosomal membrane. So these are the permeases that is present in the lysosomal membrane, helps to reuse of the components by the cell. So basically whenever these products are digested within this lysosome or autolysosome, okay, so then these components or micromolecular components are getting outside from the inside of this lysosome to outside of the cell, that means in the cytosol of this cell, and these molecules are reused by the cell. Okay, reused by the cell. So here's the definition of macro-autophagy. So macro-autophagy relies on the de novo formation of cytosolic double membrane vesicles, autophagosomes, to sequester and transport of cargo to the lysosome. Now it is the portion of second type of autophagy, that is the chaperon mediated autophagy. So whenever chaperon mediated autophagy are in formation, that means Chaperon mediated autophagy only works on unfolded polypeptides that are faultily produced by the cell. So whenever a cell trying to produce a protein, that means it initially produces a polypeptide chain. So whenever these polypeptide chains are fault in nature, then this type of autophagy comes into play. So at first, these proteins or unfolded proteins or faulty proteins have a particular motif, that is KFERQ motif. So these KFERQ motifs has bind with HSPA8, that is heat shock protein A8. So heat shock protein 70A8. So HSPA8 type complexes binds with this particular unfolded protein, and then these HSPA8 complexes helps this unfolded polypeptide to get into the lysosomes with the help of lysosomal associated membrane protein 2A, that is LAMP2A. So this red structure is specifically lysosomal associated membrane protein 2A. So whenever these HSP complex, so HSPA8, these complexes are binds with this particularly LAMP2A, that is lysosomal associated membrane proteins, then this unfolded protein or unfolded polypeptide is getting into the lysosomes, and whenever these unfolded polypeptides are moving into the lysosomes, then these lysosomal hydrolases, so these black are lysosomal hydrolases. So lysosomal hydrolases degrades these polypeptide chains and break down into amino acids, and those amino acids come outward from this lysosome with the help of these permeases, and it now present within the cytosol, and the cell is reuse these amino acids to produce further proteins. And third one is the micro-autophagy. So what about the micro-autophagy? So whenever these lysosomes are directly uptake these cellular components, that is the faulty components, then the micro-autophagy takes place. So micro-autophagy involves the direct uptake of the cargo through the invagination of the lysosomal membrane. So in this case of micro-autophagy, direct uptake of the cargo and the proteins or whatever molecules it is, that is a faulty cargo protein. So these faulty proteins or faulty components are directly uptake into the lysosomes and degraded by the lysosomal hydrolases, and it basically reused by the cell. So this is the three types of autophagy. One is macro-autophagy. When macro-autophagy is there, so macro-autophagy plays an important role in the formation of double membrane. So whenever a double membrane is formed within the cell, that is the endosome, those endosomes are directly attached with this lysosome, and lysosomes digested those products and reused by the cell. And in case of chaperon mediated autophagy, here HSP complexes are bind with these unfolded proteins, or helps these unfolded proteins to getting inside the lysosome and degrade these unfolded proteins into amino acids, and the amino acids are reused by the cell. And in case of micro-autophagy, lysosomes direct uptake the cellular intercellular components within it and digested with the help of lysosomal hydrolases, and cell reuses those components and makes another product from it. So this is the video of autophagy and types of autophagy. If you like that video, please hit the like button. Don't forget to subscribe, and thanks for watching.